Hi everyone, welcome to this new edition of SNAP Special Series. I am Alexis Murillo from the Comprehensive Center for Precision Oncology at Universidad de Sao Paulo. Liam Hourigan from the University of Melbourne, and I have the pleasure of receiving today Professor Hendrix. Dr. Hendrix is a professor at Ghent University and group leader at the Cancer Research Institute Ghent, specializing in extracellular vesicles and their clinical applications. She holds a Master in Bioscience Engineering and a PhD in Health Science, leads major international initiatives in the EV field, and received the 2021 ISEV Early Mid-Career Investigator Award for her pioneering contributions to EV research and standardization. So today, I will present getting involved in ISEV's task forces and special interest groups to shape the future of EV research. Dear Professor Hendrix, thank you for accepting our invitation. Before starting, I would just like to remind our audience that they can write down their questions in our chat box, and also then Liam will share after the talk. Please, Dr. Hendrix, the floor is yours.
Okay, so first of all, thank you very much for this opportunity. Sorry, I was seeing a message. Thank you for this opportunity to elaborate a little bit more on the science and journal concept within ISEV. So I think when Sarah came up with the idea, I thought it was very intriguing because indeed ISEV is always looking to attract early career researchers to all the initiatives that they are making. So I think this is a really nice opportunity to inform you a little bit more. And today, in particular, related to science and journals. So since a little bit more than a year, I'm acting as the interim executive chair of the Science and Journal Committee. And in this presentation, I will focus on which opportunities are there for early career researchers and how they can actually start to interact. So as a general overview, if you take a look to the ISEV website, I think you are all very well aware, being members, that it's one of the largest communities that is uniting extracellular vesicle researchers around the world. And actually, ISEV has been one of the communities that has done a lot of proactive steps towards attracting and also supporting early career researchers. And how is that? For example, by the development of MySEV guidelines. And this is not only specific to the early career researchers, but of course to any kind of researcher that is entering the field of extracellular vesicles. We are all very well aware that this minimal information guideline set is very helpful when you're entering the field, setting up your first experiments and downstream analysis and so on. Besides that, ISEV is also very active in organizing annual meeting workshops where there are always initiatives towards early career researchers to also present, participate in discussions, join workshops and so on. The EV journal clubs that are taking place online, which is also trying to be mainly focused towards early career researchers that also get an opportunity to present their most recent work towards other researchers in the field. A career center where you can explore opportunities towards future PhD or postdoc positions. And of course, multiple types of awards are also provided by ISEV to support researchers in traveling to attend these different types of meetings. But besides that, there are, of course, two other main initiatives which relate to science and journals. And here it's maybe less clear to early career researchers how specifically they can contribute to each of these specific topics, and actually also what ISEV from these 2 point views is actually looking for. So today, focus on science and journals, and I will first dive into the science part. And to do that, I know we are almost ready for ISEV 2026 in Puerto Rico, but I want to take you back to the ISEV 2025 meeting in Vienna. where I organized the educational day. And I think this cartoon was developed during the Education Day by Robert Six. And I think it gives a very nice overview on the broad scope of the extracellular vesicle field. So during the past decade, there have been tremendous advances in our knowledge on multiple different aspects. So for example, we know that there are extracellular vesicles, they are very heterogeneous, there are other types of particles, but we still don't fully understand their identity, how we are able to distinguish these different types of particles. We try to understand their biodistribution in terms of their uptake, fate, function. We start to explore these extracellular vesicles in different types of sources, which can be derived from human body fluids, but also from other environmental sources such as seawater and so on. Multiple technologies have been developed that allow us to actually very elegantly analyze these extracellular vesicles. And most importantly, building up all this know-how and biological understanding that we are developing, we are also moving towards translation, developing applications to use these extracellular vesicles, both from a therapeutic point of view, but also from a biomarker point of view. And within all of that, it has also become clear that it is very important that we report the data that we generate, because reproducibility, especially in the translation part, is of course a crucial aspect in all of this. And to do that, I want to take you back to this one slide where we once made the effort with my lab to do a systemic analysis of literature. And indeed, if you look toward literature, and I'm quite sure if you're also reading publications related to extracellular vesicles these days, and you check the material and method section, you will often find different types of methods that are used to isolate extracellular vesicles. And this really reveals that there is a lot of heterogeneity. So we have biological heterogeneity at the level of extracellular vesicles themselves that differ in their size, composition, the origin, the function, but also at the level of the methods that we use. So for example, for the 1,200 publications that we analyzed, there are more than 1,000 unique separation protocols reported. And this heterogeneity is crucial. It stimulates advances in our understanding of extracellular vesicles, but we also have to be aware that it impacts potentially reproducibility. Because in the end, everything that we are doing aims to advance extracellular vesicle research, but then we also have to understand what has actually been going on. So for that, I refer in this slide back to the wrap-up that I made at the ISEV 2025 meeting. And actually, Rink very much likes this slide. For me, he is the guru towards reproducibility in the EV research field. He is also leading the scientific reproducibility subcommittee within ISEV. And he is really taking the lead in making sure that we can connect all the different nodes and advance knowledge in extracellular vesicles. And of course, I made the reference to a symphony, into a scientific symphony, because the conference last year was taking place in Vienna, which is of course well known for his musical background, historical background. Now, to do this reproducibility effort, we also did a survey amongst the participants of the educational day just to check whether they thought reproducibility was an important aspect and a concern in extracellular vesicle research. And if we actually look to the results, so we had over 200 people participating in this survey, more than 90% actually indeed agreed that there is an existing but also an expensive problem, because of course, if we fail to reproduce experiments, we waste a lot of research budget and also, of course, valuable time. So as a rapidly advancing and growing field, it is crucial to also pay substantial effort towards building efforts that can help to stimulate reproducibility in the field. And actually, ISEV has been very supportive towards this reproducibility aspect. So already the very first, we didn't really call them ISEV guidelines, but there were already first manuscripts in 2013 and 14 that already outlined some guidelines and some issues that researchers may consider when working with extracellular vesicles. And this then further developed into the ISEV 2018 guidelines and the 2023 guidelines. But alongside that, ISEV wanted to take this effort one step further, and they decided to establish a rigor and standardization subcommittee in 2019. And here the aim was really to drive efforts that for dedicated particular fields want to develop a set of guidelines to further enable reproducibility in these different subfields. Now, between 2019 and 2024, there have been, of course, a lot of discussions going on reproducibility. And originally, the naming of this subcommittee was rigor and standardization. And we started to discuss that maybe standardization is not fully what we are looking for. What we are looking for is reproducibility. We want to be able to reproduce what others have been doing, which is a critical step towards translation, whereas standardization more indicates that we aim that everyone is doing exactly the same, following exactly the same steps, the same protocol, and so on. And this is actually not what we aim for. So we decided to change the name from the Rigor and Standardization Subcommittee into the Scientific Reproducibility Subcommittee. And this brings me immediately then more specifically to the science part. So this Scientific Reproducibility Subcommittee overlooks two important aspects. And these are the task forces and the inter-society working groups. So in the next few slides, I will explain a little bit what the aim of these different groups is and how early career researchers can, of course, also contribute to these initiatives. So let's first have a look to the task forces. So since the launch of this initiative in 2019, there are actually currently 15 task forces that are up and running. Now, what are task forces? Task forces are expert groups that are established to strengthen the reproducibility of EV research. And if you take a look to the different topics of these task forces, you can observe that they really focus on a particular biofluid, like blood, milk, solid tissue as well, on some biomolecular coronas, on some biomolecular components such as RNA, but then also initiatives towards translation. What are the next steps if you really want to translate vesicles into applications with a task force on regulatory affairs and so on. But these are really dedicated groups that focus on one particular aspect, have certain defined goals that they want to achieve within a certain time frame. So what is the purpose? They really aim to strengthen the rigor and the reproducibility of EV research, and they really focus on setting methodological standards that can help to improve reproducibility. They address particular challenges, like, for example, when working with blood plasma, what are now the pre-analytical factors that can have an impact on the downstream extracellular vesicle analysis? They develop a community consensus. So think about, for example, the urine task force, but also the blood task force that have developed some guidelines that can help you to initiate experiments with when working with these biofluids, especially considering from the aspects of biobanking, pre-analytical factors, and so on. So they really work on very defined topics. Now, what type of activities do these task forces undertake? So typically they work towards a set of publications, so where they meet with a group of experts that are trying to generate a consensus and share this consensus with the research community. Often these outcomes in terms of consensus that is achieved on a particular topic within this task force is also, for example, becoming integrated in the MySEV guidelines in a later stage. Also education and outreach. So they also try to develop webinars, but organize as well journal clubs with the aim to share the consensus towards, of course, achieving reproducibility. The more people are aware about the aspects that they consider as experts to be relevant, the more beneficial it would be to also for the community to have access to it. So beyond publications, they often also organize educational sessions and outreach sessions. And to develop this consensus, although they often have a discussion within the expert group, commonly they will reach out to the ISEV community by means of surveys, for example, to also collect the input on this consensus development, but as well they can organize workshops and so on. So what really the added value is of these task forces is really aiming with a particular focus to increase reproducibility and transparency by defining best practices and sharing this information by different types of products, as I mentioned, meetings, publications, and so on. And they also help and try to align, of course, then researchers across different disciplines. And ultimately, of course, as I mentioned, they aim to implement these guidelines then into the MySEV guidelines, and by doing that, really impact, but most importantly, strengthen the research on extracellular vesicles. So when we look in particular to task forces, how can early career researchers participate? Well, there are actually multiple ways that can be explored. The most important one is join the activities that are being organized by these task forces. So if they send out surveys, provide your input to these surveys. If they organize workshops, try to apply to also attend these workshops and webinars, especially when they are online. Respond also to calls for task force members. So often when new task forces are launched, there will be an open call to the ISEV members where you can actually apply to become a member of the task force. One important note is that often the task forces are limited in their number of participants. So most of the task force consists of expert groups of 20 to 30 people. So there will always be a selection because most of the time there is a very high interest to join these task forces. Nevertheless, we have also recently updated the SOP related to task forces, so the governance of task forces, where we have also indicated that the participation of early career researchers should be definitely considered and also implemented. But beyond waiting for a new call for task force members, also think about actively reaching out to the task force chairs. Like if you have developed an interesting manuscript or generated important data that relate to a particular task force, don't hesitate to directly reach out to the task force and ask whether you are able to also join based upon the recent experience that you gained, and they will very likely be very welcome to have you join the task force, or at least share by means of a presentation, for example, the results that you obtained, so you can participate to the discussion. And of course, there is also the possibility to propose new task forces. So on the ISEV website, we now also have made a survey link where you can propose a new task force topic. Now, as an early career researcher, it is less common that you will be able to share the task force, although we have a recent example where actually early career researchers related to core facilities are actually sharing a task force. So there are definitely possibilities. If you think this is a very interesting topic, we need to build a consensus, we need to group experts to provide some guidance on this topic, you can definitely propose as well a new task force with envisioned task force chairs and objectives, of course, as well. So to conclude on the task forces, actually, it's one of the most direct ways to contribute to the EV community. Also at the ISEV meeting, at the annual meeting, most of them will actually have scheduled a satellite event where you can immediately participate into the discussion and also indicate your interest to become a member of this task force. And actually many of the guidelines and resources that we use today as a guidance originate from these collaborative initiatives. So as I mentioned, many of the outputs are in the end also integrated into the MySEV guidelines. Besides the task forces, the Scientific Reproducibility Subcommittee also overlooks inter-society working groups. Now let's have a look to what these working groups are. So the big difference is that inter-society working groups actually want to establish connections with other types of societies that have a same common interest. And the main aim of these inter-society working groups is actually making sure that the knowledge that has been developed in the task forces also becomes visible in these other societies that also have a keen interest in extracellular vesicles. Now, to give one example, or at least the 3 inter-society working groups that are currently established is between ISEV and the Liquid Biopsy Society, a Cell Therapy Society, Cytometry Society, and Thrombosis and Hemostasis Society. And what is often being done is that there are, for example, common symposia that are organized, also satellite events during the annual ISEV meeting, where these societies meet, interact, and also exchange knowledge, because, for example, ISEV is, of course, very dedicated towards building knowledge on extracellular vesicles, whereas, for example, ALPS is more from a clinical point of view, advanced in how to translate biomarkers into a clinical application. And by merging the two, ISEV can learn on how we can translate our biomarkers that are EV associated into a clinical application, and what can be the barriers that we need to overcome. But on the other hand, clinicians that are involved in the ALPS also become aware about the minimal information guidelines and the aspects that you need to consider when you want to study extracellular vesicles in biofluids. So the purpose is really exchange expertise and promote the best practices. Connect the researchers that have complementary scientific interests and make these other societies or spread the outputs of all the hard work that is being done by task forces. And that, for example, results in the MySEV guidelines, but also the position papers, any reporting standards, that they also gain visibility beyond the ISEV society in the more broader scientific communities. So the activities that these inter-society working groups undertake are often joint position papers, dissemination of the ISEV guidelines, joint workshops and discussions, which can be as a satellite event at the annual ISEV meeting. But most often, these working groups also have additional meetings that they co-organize with their cross-society partner. So again, there are multi-opportunities here, also for early career researchers to participate to these meetings and contribute to these discussions. So what is then the added value of these inter-society working groups? Well, I think I already explained it a little bit on the first slide of these inter-society working groups. But for example, we bring the partner societies our know-how on extracellular vesicles, while these partner societies bring additional expertise in the field of clinical medicine, flow cytometry, cell therapy, and so on. So there is a little bit of a mutual benefit that helps to advance different fields moving together, in the end, of course, aiming to advance extracellular vesicles and their different types of applications. So how can you participate here as an early career research? Basically, it comes to the same end result as with the task forces. First of all, participate in the activities. So often these inter-society working groups also distribute surveys that seek for your input. And I know sometimes a lot of surveys end up in the email, but this is really because consensus building can, of course, only occur when also the community is participating. So whenever you see these surveys popping up in your inbox and you have the relevant expertise to answer to these surveys, definitely try to do so. And actually often by also making additional comments in these surveys, the chairs of these working groups will pick it up and may even also get into contact with you for any of the ideas that you have shared through these surveys. Participate in symposia, satellite events. Also, the cross-society initiatives, of course, can be attended. Webinars or online meetings are often also organized by these working groups. And the main aim is to really partner with the other societies. So a little bit to understand then, what is now the difference between an inter-society working group and a task force? So a task force really has a dedicated timeline, so most of the time 3 to 4 years, with a group of about 20 experts that discuss on one particular topic, and they aim to end with a certain product, which can be an educational tool, it can be a guideline, it can be a consensus, it can be a webinar. Whereas for the inter-society working groups, this is of course slightly different. They act in concert with other scientific communities where they really want to share expertise. And basically, as I mentioned, task forces do a lot of hard work in developing guidelines. Well, the main task of these inter-society working groups is spread the word to these other scientific communities. So the differences between task forces and inter-society working groups really lay in the duration. So of course, inter-society working groups are less limited in time compared to the task forces. Does it mean that all task forces are restricted to 3 to 4 years? No, of course not. Blood, for example, the Blood EV task force was already established in 2019 and it's still ongoing today. And why is that? Well, because there is a high interest in studying blood as a source of extracellular vesicles, but there are also still a lot of unaddressed needs in that particular field. So there is, of course, a transition in the leadership of these task forces, and there is always a new set of objectives that are being established that should be achieved within a certain time frame. But it's more really with a clear agenda, clear deadlines, and we should achieve this aspect within the next 3 years. For the inter-society working groups, because it's of course aiming at establishing connection with other societies, this is less limited because of course also the other societies will continue to exist, to grow, and so on. But of course, the field rapidly expands. It means that it may be interesting for ISEV to connect with other societies beyond those that are currently established. So if you think you're working in a field and you think it would be really good to interconnect ISEV with the other societies, again, make the proposal, reach out to any of the chairs, to me, to other people from ISEV to discuss this, because we are very open to that. And the same for task forces. So in terms of science, I now discuss task forces and inter-society working groups. And then we also have the special interest groups. Special interest groups, they don't fall under scientific reproducibility subcommittee. Why? Because their main focus is not reproducibility, but is really science. So special interest groups are thematic communities that foster collaboration and knowledge in specific EV research areas. And here I have actually visualized both special interest groups that have currently been established, which is EVANS, extracellular vesicles in neuroscience, and then GUSEV on the EVs in the genitourinary system. So these are currently the two special interest groups that exist. So you immediately observe one on neuroscience, one on the genitourinary system. So a dedicated scientific area from a research science point of view. So they want to work together with other societies on this particular scientific topic. Special interest groups, in contrast to the task force and the inter-society groups, they don't have a member restriction. So anyone who is interested to join these groups, they are welcome to join these groups. So I think currently EVANS and GUSEV have 200 to 300 members that attend and participate to their activities, including a lot of early career researchers as well. So as I mentioned, the purpose of these special interest groups is to connect researchers that have interest in the same type of disease, same type of application. So they really want to promote exchange and collaboration from the science point of view, less from the reproducibility point of view or the technology point of view, but really from the science point of view. So what are the activities that they undertake? Again, of course, like with any of the other groups, they organize a lot of meetings, also contribute a lot of the journal clubs, have seminars, have of course joint meetings with other societies like neuroscience societies and so on. So again, it is a sort of community building, but now from a very scientific point of view, focusing on a particular research area. And now neuroscience and the urinary system, GUSEV, they often organized and recently actually had multiple in-person meetings, which they joined together with other societies that focus on this area, but they also have very successful online webinars with also high participation of early career researchers. So they really want to strengthen collaboration from scientific point of view and generate new research directions, focus on emerging topics and questions that really remain from a scientific point of view. So how can early career researchers participate here? Become a member. First of all, if you're working on that particular topic, so I mentioned now we have two special interest groups, There will be 2 new special interest groups upcoming very soon. So if one of these special interest groups lays within your field of interest, definitely become a member. It will be a very valuable group for you and to contribute to as a researcher, but also to learn from the expertise, of course, from the other experts and researchers that are present in this society, in these subgroups. Participate again to the surveys, the journal clubs, the symposia. Again, they also organize satellite meetings. And this is really a way to connect with researchers that work on the same research domain. And besides that, task forces, inter-society working groups, and special interest groups, they also interconnect together. Indeed, for example, if we look to the GUSEF special interest group, they very closely work together with the task force on the urinary EVs, which means that all information consensus that is built within the task force of urinary EVs is immediately also spread through the special interest groups to the other connected societies to the special interest groups. So special interest groups go really beyond ISEV and also attract members from outside ISEV that have the same interest in a particular disease, for example, from an EV point of view. After that, let's now move into the journals. So I think you're very well aware that we have 2 types of journals that have developed by the ISEV community. So first of all, we have the Journal of Extracellular Vesicles, which is the flagship journal from the society, which is really centered towards extracellular vesicles. Besides that, there is also the Journal of Extracellular Biology, which is a little bit of a broader scope compared to the Journal of Extracellular Vesicles and has in general interest into the extracellular biology and communication, so it moves beyond other types of particles and molecules that can be found in the extracellular environment compared to EVs. So, of course, the main aim of these journals is for ISEV to be able to support the EV research community. And both journals aim to disseminate high-quality research and advances in EV science. So their main focus is, of course, publish articles, which can be original articles, can also be review manuscripts. Think also about the MySEV guidelines, position papers, consensus documents that are established by the task forces that result from some of the workshops that have been organized. These are all products that commonly are published by these journals. The aim is, of course, that they really want to disseminate and provide visibility to an EV community. So if you have interesting work available that relates to extracellular vesicles, definitely consider to also publish your work in these 2 journals. From my own personal experience, I would say that sometimes you write a manuscript and you really have the idea, this is a manuscript that is really of main interest to the EV researchers, then definitely submit it to one of these journals because you will reach the correct audience. If you would publish it to another journal that has a much broader scope, it may receive less attention or maybe less reach the community that you're really looking for. So when you're writing down a manuscript as an early career researcher, always reflect on which journal do I have in mind and where do I think this message would be best delivered if it is in an EV type of area community you're looking for. These 2 journals are definitely the place to consider and to be. So what is the added value? providing visibility, very strong visibility within the EV community. But both journals actually also pay a lot of attention to ensure rigor, transparency, and reproducibility, that these aspects are well addressed within the manuscripts that are being published. So how can you participate as an early career researcher? Of course, submit your best original research article. You can also write reviews that can be considered for publication. An important topic, participate in peer review. It's only by contributing to peer review that you understand how reviewers are evaluating a manuscript, and that helps to improve your own manuscript writing, how you prepare a publication for submission, and so on. Many of the manuscripts that are also being published in one of the journals are also discussed during the online journal clubs. And the journals also hold satellite events. So if you want to have a direct interaction with the editors-in-chief, or whether you want to check is your publication suitable for this type of journal or not, if you attend the annual ISEV meetings, definitely make sure to also attend the satellite event from the editors-in-chiefs from both journals, because they will give very valuable information and also try to support you in that. So together, both Jeff and GX Bio ensure that EV-focused discoveries and the broader extracellular vesicle communication field in general have a very dedicated platform within the ISEV community. So with that, I think I almost am at the end of my presentation, and I want to conclude by summarizing the 5 ways that SNEF members can engage within the ISEV science and journals. The first way, I think the most simple one, if there is a special interest group that covers your scientific interest, join it. Join it as a member, connect with the researchers. Will be early career researchers, there will also be experts, but it is a subgroup of 200, 300 people, and especially in smaller groups, establishing collaborations is much more smooth than if you're in a much broader community. So also stay tuned, because new special interest groups will be launched in the upcoming year that maybe cover your interest, and then definitely consider to become a member. The membership is free actually for ISEV members. In these groups you can discuss, you can set up collaborative projects, and it will definitely advance you from a scientific development point of view, but of course also your scientific projects as well. Participate in the task forces. So if there are any opportunities to participate in the task force, first of all, if it is through participation in surveys, to online events, satellite events, if they are within your interest, please contribute to them, because it's by contributing and showing your interest that in the end you will be actively involved in participating in these task forces. Then, of course, share your research with ISEV journals, and also try to contribute to the peer review. If you're not yet listed, but if you're interested to contribute to the peer review, I think in the ISEV newsletter that is often circulated, you can also, there is a link if you're interested to become a peer reviewer, where you can complete a survey, and then you can be considered or you can be requested to review certain manuscripts. So that's something you can also explore. And then participate, of course, in the ISEV meetings, in the workshops. So it's really through collaboration, establishing connections, that you get integrated into these aspects. And I noticed there was a SNEF presentation before by Randy Corney on how to why to attend a meeting and how to get most out of your meeting. Well, it's really through interaction. So really interact, ask. If you don't ask, the answer is anyway no. So you can only try to gain. If you're interested in joining a task force, try to meet with the chairs during any of the meetings, send them an email and try to convince them why it is important for you to contribute and which experience you can bring to make this task force successful, for example. So in conclusion, I think it's very important as early career researcher to realize that ISEV is really a community-driven society. So its impact also depends on the active participation from researchers like you, early career researchers. So with that, I want to thank you for your attention, and I'm happy to address any questions.
Thank you, Anne. I think we can all agree that was a pretty thorough and very insightful look at all these different kinds of organizations in the EV field. Thank you. And yeah, as you mentioned, it's important to actively contribute. It is community driven, both SNEV and ISEV. So this is a good chance for anyone to ask any questions they might have of Anne. So right now we've only got one in the chat, and that's from Murillo. And they are asking if there is any common group or mail ID where early career researchers can share their experimental problems.
Where they can share their experimental problems. Well, of course, each of the different groups would be good to address, but I'm not sure anymore. But I think on the ISEV website, there has been a sort of a communication group where you can start to chat. But I think to address any of the experimental comments, reach out, check in literature who may be around that has this expertise, directly email to the authors. Most of the time, they are very happy to help you out. And besides that, if you don't know or your answer is not being addressed, in meetings also reach out. Like, I come from the same situation. I also once was an early career researcher, and I was a little bit shy and afraid of reaching out to people, but really don't be. I can tell you as well, I'm also happy now. I'm a little bit more grown up and not at the early career stage anymore, but now I understand it's no problem. Why wouldn't we want to help? So if there are issues, experimental problems, try to find out who are the experts in the field and reach out directly to them. Don't be hesitant on that at all. Most of the time they will be very happy to help you or to forward you to the postdocs or the PhD students from their team that may be able to help you as well. And I think actually SNEF would also be a good group to actually discuss some of these experimental issues because most of you may, of course, sometimes struggle with the same experimental aspects. So I think also discussing this within SNEF is, of course, also a possibility.
All right, if anyone else has any questions, please just raise your hand. So next up we have Sarah.
Thank you, Liam. I just want to make a quick note on what Anne was saying. So SNEV actually has a Discord channel where we had a place where people can go and ask questions, and someone from the community can have had a similar issue or similar protocol or experiment, so you can reach there. The Discord channel has been a bit shushed, like a bit silenced. People haven't engaged as much, but there has been a need by other people as well to have a platform where people can like communicate. So maybe we can just need to bring that up a little bit. But my question for Anne, actually, Anne, this was amazing talks. Thank you so much for the work you put and like presenting all the options that ECRs can volunteer. There's so many opportunities, and I feel that people sometimes don't know about them. And my question kind of comes from, I think that ECR sometimes are a bit more like shy. They might be like, oh, I'm only a PhD student. What do I have to contribute for a SIG or a task force, or I've only just started? So what would your message be to those people? Is it still worthy for like a 1-year, 2-year PhD student to join a SIG, because they can still learn something even if they can't contribute as much? So what would you tell like people that might want to join, but they feel they don't have that much to contribute? So what would you say to those people?
Well, in general, as I mentioned, so task forces are a group of experts. They really also have a dedicated timeline, so they have to make their or accomplish their achievements within 3 to 4 years. But what we now changed is that we really like new task forces that are being established or when memberships are being renewed. So there have to be transition phases. Like people cannot stay on there, like the chairs are only there for 3 years. So there is a very rapid renewal. But actually for the new task forces, we stimulate that early career researchers are really part of the task force. And then I can only recommend, if any of the new calls come out and it falls within your field of interest and in the area that you're working, apply. Because if you don't apply, it will be very difficult to step in at one point. So especially for the first, second year PhD students, I think that would be a very good way. And then also explain what you can contribute, and don't be too shy on that, because although you may feel less an expert in that topic compared to people that have been there for 10 years, it doesn't mean that you don't make valuable contributions, because often you bring actually very valuable information because you have the hands-on issue. You are really encountering it from a lab point of view, which many of the experts are really not really in the lab anymore. So I think it's really of a additional benefit. And also many of the task forces, they also first make an overview of the current state of the art by exploring literature. And I think anyone is capable of doing a literature analysis, and this is definitely a type of task where early career researchers can be very, very helpful, even in the task forces. So there are different types of options that you can propose, because most of the time, if these surveys are launched, they will ask, what can you contribute to the task force? So very well reflect on how you want to address this question, because you cannot just be in a task force to be there. Of course, you have to make an active participation. So think about how you see your role within the task force. It can be indeed to explore literature, or from your bench expertise that you can contribute some insights. So yeah, really give it a try and well reflect on how you position yourself functioning within this task force. And for the EV-Track task force, this was actually a special one, and there the majority of people participating are actually early career researchers, because we were looking to really involve the research level that is actively submitting manuscripts to EV-Track, so that they know how the workflow from an admin point of view is going on, and so on. So there will definitely be increasing opportunities for early career researchers to participate, but of course you have to apply. So if you see any of the surveys popping up in your email, really reflect and try to make a good proposal.
Thank you, Anne. That was very helpful. Thank you.
You're welcome.
And next we have Yetem.
First, thanks so much for the talk. It was I just get so many new information. I have basically two questions. The first thing is, regarding to the ISEV journals, as we know that we are time bounded because as a students we have the time that we start and finish, and would it be possible to know the time it takes for the publication, like from submission up to the publication with the ISEV journals? And the second question is because I am working in a small group, and then I'm the only one who's working with the EV. So I believe that having journal clubs is really important. And then I was participating with the SNIP journal clubs, but I don't know if they are existing. Maybe I missed the emails. And do ISEV do have journal clubs that we can actively participate? So these are my two questions. Thank you.
Yes, thank you for the question. So first of all, the timeline for publication. I'm not going to put a very concrete time window on that because it depends on a lot of different steps. But well, I think there are two important considerations. First of all, with the journals, we are working hard to make sure that we can keep this timeline between submission and final decision, acceptance or rejection, can be as short as possible. And that's actually why we also need early career researchers, because you know what the most difficult part is for us within the journals, the rate limiting step, is actually the difficulty of finding reviewers to evaluate the manuscript. Sometimes we invite 10 reviewers and none of them are available to review the manuscript. So this is really an issue that not only the ISEV journals are currently encountering, but also other journals. So again, be aware, it's important also for early career researchers to take this opportunity to be involved in a peer review process. There have been some delays both for GXBio and Jeff, which has to do with the transitioning between the different editorial teams. So for example, for Jeff, we had a switch from the previous editor-in-chief to a new editor-in-chief team. And this, of course, comes with a slight delay. But currently, all editors-in-chiefs from both journals are really making a lot of efforts to keep these timelines as reasonable and short as possible. So we will do our best. I'm in the same situation. My PhD students, they also have to submit their work before they are able to defend their PhD. But I think in the majority of cases, these manuscripts go through a very smooth pipeline. There are a few exceptions, but I think this happens in any journal, and it's not in particular related to the ISEV journals. And for your second question, whether there are any journal clubs, so SNEV has journal clubs, but we have the EV Journal Club YouTube channel in general, which is mostly on the Wednesdays in the late afternoon in European time frame. And there you can actually reach out yourself. If you think there is a paper which would be good to discuss, you can definitely reach out to the organizers of this EV Journal Club. So they do active solicitations to check whether people are willing to interest, but you can also propose yourself. And these always become available on the YouTube channel as well. So you can always catch up with any of the journal clubs later on.
Thank you so much.
You're welcome.
Does anybody else have any questions before we wrap up? Okay, well then, thank you, Arne, and thanks everyone for your questions. This is the end of the presentation.
Then I would say thank you if you give me the chance, and I look forward to see you all contribute to any of the initiatives in the near future.
Thank you so much, Anne. Bye, everyone.
Bye.
Thank you.